In this video, we're going to study abnormalities that can occur with meiosis. The idea is that divisions that we saw, like mitosis, meiosis, are not perfect. And if there are problems with these divisions, then problems can occur in the organism. So the goal of meiosis is to make gametes. If the gametes are missing chromosomes or have extra chromosomes, then that means that the organism that they make, the zygote they make, will be missing chromosomes or have extra chromosomes, and that will lead to certain developmental disorders, which we'll discuss in a bit of detail. We're going to see how that happens at the level of meiosis. We're going to take a look at karyotypes, which can be used to see if you have any chromosomal abnormalities. We'll take a look at ways that you can check for non-disjunction if you're an expecting parent. And then we'll take a look at some reproductive technologies. We won't spend too much time talking about these. These I would want you to read up on your own, and they are fair game for a test. You can add these to your notes. So you can talk about artificial reproductive technologies, such as in vitro fertilization. There's a few others you can read up on in your textbook, and then add them to your notes, just a brief summary to know briefly what they are. So once again, remember that to become you have to be made from sperm and egg. So you had these gametes. There was a sperm with 23 chromosomes and an egg with 23 chromosomes, and when it combined, it made, well, you with 46 chromosomes. And so you had the correct number of chromosomes, and mitosis occurred with no problems, and then you became you. But sometimes there are problems with meiosis, and as a result, the gametes produced have some abnormalities in them, which then get passed on to the zygote that they make. And so we're going to take a look at what happens if you have too many chromosomes or too few chromosomes, or if even the chromosomes are damaged. We'll take a look at what happens there and what type of disorders can occur. So this picture is a karyotype, and a karyotype is just a picture of your chromosomes in a somatic cell. The reason I know it's a somatic cell, just looking at this at first glance, is because there's 46 chromosomes, or 23 pairs of chromosomes. So what I want to point out is that there's 22 pairs or 44 chromosomes called autosomes. So that would be 1 all the way to 22, these pairs here. And they have genes on them that control everything but sex. So they might have genes for eye color somewhere. There might be a locus for eye color over here, and then a locus for eye color over here. One of these was from mom, one of these was from dad. You might have a locus for, I don't know, hair color over here, and a locus for hair color over here. One of these was from mom, one of these was from dad. But the idea is that these autosomes, these 44 autosomes, have the genes for everything but sex. And then lastly, there's a pair of sex chromosomes. That's the last pair that we put in the karyotype. And those chromosomes, those sex chromosomes, control your sex. So if you have XY, that means that you're a boy, and if you have XX, that means you're a girl. Mothers can only give X, so whenever a baby's made, the mother will automatically give X. What really is going to determine if you have a boy or girl is what the father gives. The father can give X or Y because the father has X and Y that it could give. So if the father gives a gamete with Y, then you'll get a boy. If the father gives a gamete with X, then you'll get a girl because the mother had no choice but to give X. So here we can see the karyotype of a female, and I know that because there's 2 X chromosomes. And here we can see the karyotype of a male. I know that because there's a Y chromosome there. Notice the way the chromosomes are arranged. They start with the longest pair and end with the shortest pair over here for the autosomes, and then we have our sex chromosomes that follow. So you'll notice, for example, that we have 5 columns, and essentially we go from biggest to smallest in our rows, biggest to smallest, biggest to smallest, and then biggest to smallest, with our X last. And that's typically how they're arranged. Remember that the pairs are homologous pairs. One is from mom, one is from dad, and that they have the same gene loci, the same length, the same centromere position. That's the case for most of the homologous pairs, with the exception of the X and Y. They're still homologous, they just don't have the same length, they don't necessarily have the same loci, but they do have some similarities because they do pair up during prophase 1 of meiosis. So the karyotypes that we took a look here were normal karyotypes. They had 46 chromosomes, and there was nothing missing, there was nothing extra. They were just a normal karyotypes made by most likely normal gametes, so normal sperm and a normal egg with 23 chromosomes each to give 46 total. Now, let's take a look at the karyotypes on this page here. So usually what I do is I like to count the autosomes. And if you look at the autosomes here carefully on this page, or on this karyotype here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and then a pair of sex. This one seems like a normal individual. There's 46 chromosomes. You could even figure out that it's a boy, a male, because of the XY. Now let's take a look at this karyotype over here. You'll notice it's a similar arrangement to what we saw earlier. We have most likely a female over here. But if you look very closely, if you analyze this very closely and look at chromosome 21, we are diploid organisms. We should have 2 of each chromosome, but you'll notice here we have 3 of each chromosome. So this is an abnormal karyotype. So this individual has an abnormality with chromosome number 21. So we number our chromosomes, again, longest to shortest, longest being 1, shortest being 22, and then we have our sex chromosomes. But when we analyze these karyotypes, we need to see if they're all paired up. And all of them are paired up except for chromosome 21. So we have our human male karyotype, and our female karyotype over here. But you'll notice that, again, because we have 3, that's an abnormality, and we call this abnormality trisomy. Trisomy is having an extra chromosome in a certain location. So here we have trisomy, specifically 21. So trisomy 21 would be the name that we would, genetic name that we would give this disorder. However, most people would know this disorder as Down syndrome. So whenever you have extra chromosome 21, genetically we call that trisomy 21, but the other term that we would give this condition is Down syndrome. So again, the idea is that sometimes you might have chromosomal abnormalities. The abnormalities could be in terms of the structure of the chromosomes or in terms of the number of the chromosomes. So in terms of chromosome number, that basically means you have too many or too few chromosomes, like the example we saw in Down syndrome with the extra chromosome 21, the trisomy. There are also cases where you might have too few. So you might have one missing chromosome, for example. Chromosome structure can also be an issue. Sometimes you might have the correct amount, but maybe some of the chromosomes might be damaged. So there might be a little part in your chromosome that's missing a gene, or the genes have been flipped in some way, but there's also possibilities to having issues with chromosomal structure. So the idea is that if you have any of these chromosomal abnormalities, then you can have a developmental disability, such as Down syndrome. So here we have trisomy 21, and over here you can see a picture of an individual with Down syndrome. So what causes this abnormal chromosome number? Well, here we have 2 pictures. On the left side you can see normal gametes, 23 and 23. When they combine, they should give a zygote with 46 chromosomes. Whereas here you can see that we have a gamete that has an extra chromosome inside of it, 24. And so when these combine, you'll get a zygote with 47 chromosomes instead of 46, the normal amount. And this could be an example of a trisomy. So abnormal gametes usually result because of something going wrong with meiosis. And the errors that occur during meiosis, they're called non-disjunction. So when you have a non-disjunction error in meiosis, that can lead to too many or too few chromosomes, and also some damage. But we're going to talk about the cases of too many or too few chromosomes. So again, here we can see the correct number of chromosomes in humans. We can see 23 chromosomes in sperm, 23 chromosomes in the ovum, and that gives us 46 chromosomes. You should be able to identify normal karyotypes from abnormal karyotypes at this point, and also be able to identify whether you have a boy or a girl based on the sex chromosomes, and you should be able to determine where the autosomes are. Where the autosomes are, and where the sex chromosomes are. So if you have abnormal number of chromosomes in the gametes, like I said, it can result in extra chromosomes or missing chromosomes. So you might have, let's say if you have an extra chromosome, 47 chromosomes in your zygote instead of 46. If you have a missing chromosome in the gamete, well, you might have 45 instead of 46. And so you can see this example over here. So when the error is made in the zygote, well, the zygote will go through mitosis, and then that error will be replicated throughout mitosis. And so all the cells made will have 47 chromosomes. Over here, if we have 45 chromosomes in our zygote and then it goes through mitosis, that error again is copied through all of the cells. And so now we have an organism where all the cells have 45 chromosomes each instead of 46 chromosomes each. So let's talk about non-disjunction. It can happen in a few different places during meiosis. So let's analyze this picture over here. Here we have a non-disjunction error occurring during meiosis 1. The homologs were paired, and you'll notice that the two on the top, they separate properly. They go to opposite sides of the cell. But the two at the bottom, they don't separate. They end up going to the same side of the cell. So now what happens is that in one of the cells you have an extra chromosome, and in the other cell you have a missing chromosome. So once these cells go through meiosis 2 normally, assuming everything's fine in meiosis 2, you're going to have some gametes that end up with extra chromosomes, n plus 1, and on the other side where you had the missing chromosome, you're going to have, well, missing chromosomes, so n minus 1. In other words, if you look at this cell here, there's 4 chromosomes to begin with. So you would expect that in each gamete you have 2 chromosomes. You'd have n is equal to 2, but we don't have that. We have 2 plus 1. We have 3 here. It's 2 plus 1. We have 3 here. We have 2 minus 1. We have 1 here and 1 here. So if this gamete is used during reproduction, then that leads to an extra chromosome in the zygote. But if this gamete is used, then that leads to a missing chromosome in the zygote. And so this same concept applies to any number of chromosomes. So for example, if I had 10 chromosomes to begin with, I would expect 5 in each gamete, but instead I'd have 6, 6, and 5, and 5. So I'd have the n plus 1s and then the n minus 1s produced. And you can apply that to the human number, 46. You should have 23, but you'll have 24 and 22 instead. So you should be able to practice this with any number and understand that this is non-disjunction that happens in meiosis 1, where the homologous chromosomes fail to separate. So the homologous pairs fail to separate appropriately. So this specifically was an anaphase 1 of meiosis 1. Sometimes the error can occur in anaphase 2 of meiosis 2, and we'll take a look at that in just a bit. So here we have an error that happened in anaphase 2 of meiosis 2. So meiosis 1 happened perfectly. The chromosomes, the homologous pairs went to both sides as expected. And then what happened is we get to anaphase 2 in meiosis 2, and an issue happened. The spindle fibers, they tried to separate the sister chromatids, and they did up here, but down here they didn't. So what happens is now you're going to have one of the cells over here with an extra chromosome. And this cell from this part will have a missing chromosome. So you'll have a gamete that's n plus 1, an extra chromosome in it, and a gamete with n minus 1. Now, hopefully, if the other cell went through anaphase 2 normally, then at least you'll have some gametes that are normal. You'll have your N and your N. So when an error occurs through non-disjunction, meiosis 2, you at least have the opportunity to get some normal gametes that are produced. But again, the idea is we have 4 chromosomes here. We expected 2, but because of this non-disjunction error in anaphase 2, we get an N plus 1, which is 3, and N minus 1, which is 1. And then we do get some of our normal gametes as well. So you have 2 out of the 4 gametes that are normal, which means you have a 50% chance of using one of those normal gametes, assuming you produced 4 gametes, right? Remember the difference between spermatogenesis and oogenesis. So again, just like with the previous example, you should be able to figure out what your gametes would end up with. So let's say for humans, we have 46 chromosomes. We would expect 23, but we would get some that have 24, some that have 22, and some that have 23. So we would have some normal ones. Now the idea is that, let's say we use one of these abnormal gametes, so we'd have n is equal to 24, plus our n is equal to 23. Let's assume the other parent gave a normal gamete. We'd then have 2n is equal to 47. Now, we would usually write this like this. We would say 2n plus 1 is equal to 47. That's to say that we have our 46 from our 2 sets, but then we also have an extra one. If you add 1 missing, you do 2n minus 1 is equal to 45, because a normal 2 set should be 46, but if you're missing 1, you just do minus 1. So at this point, you should be able to label where these errors are occurring, and you should be able to label what type of gametes you have produced. So here we have 3 situations, A, B, and C, and you need to tell me if the gametes are n, n plus 1, or n minus 1. So what I would do if I were you is pause the video and then try it out for yourself to see if you can answer these questions, and then once you're done, then you can come back and check your answers with me. So I'm going to start answering these questions now. We have 2 chromosomes to begin with, so I would expect that at the end of this, I have 1 chromosome in each, and it looks like I do have 1 chromosome in each. So that means that these are n. They're n. They're normal. There's not plus 1. There's not minus 1. And it looks like this was a normal meiotic event. There was normal meiosis 1 and normal meiosis 2. Great. Now, we can call this one normal. Let's go take a look at our B and our C. So we look at B over here. We have 2 chromosomes, so we would expect 1 in each. But we don't have 1 in each. We have n plus 1, n plus 1. We have 2 in each here. And then we have missing altogether, so n minus 1, n minus 1. So this looks like it was an error in meiosis 1. So this is our error in meiosis 1. The homologous chromosomes did not separate during anaphase 1, so they all ended up in 1 cell, and 1 cell did not get any at all. Over here, again, we have 2 chromosomes. We'd expect 1 in each, but we don't have that. We have an n plus 1. We have 2 here. We have an n minus 1, 1 missing, and then we do have some n's, some normal ones as well. So when you have the possibility to get normal ones, what that means is that the error occurred in meiosis 2, during anaphase 2. So you can see here that meiosis 1 happened just fine. The homologous chromosomes separated. But if you take a look at anaphase 2, there was a non-disjunction event that happened here. Here it looked okay because the 2 chromatids went to both sides, as you can see here. But here, the chromatids didn't separate and only went to one side and not the other. So this was an error during meiosis 2. So you should be able to figure out what type of gametes will be produced based on what you see in the meiosis process. You should be able to tell me if there's a non-disjunction happening or if it's normal meiosis happening. So if I were you, I would pause the video, try this out yourself, and then see what gametes are produced based on what you see in the meiosis process, and then come back and see if you have the right answer. So hopefully you have drawn something out and you made a little guess, or you have a good idea as to what the gametes should look like. Let's take a look. Over here we have 8 chromosomes. Sorry, we have 1, 2, 3, 4, 5, 6, 7, 8. That's right, 8 chromosomes. So we would expect 4 chromosomes in the gametes to get a healthy N gamete. But let's take a look, see if everything's going to plan. So here we have a correct division of the chromosomes, and here we have a correct division of the chromosomes, and here as well. So this looks like some healthy meiosis, which means that we're going to get our N gametes, our normal gametes, with 4 in each. Right? These 4 might go here, and these 4 might go over here. Same idea here, 4 in each, 4 in each. And you should be able to do this for any number of chromosomes, whether it's 8, whether it's 16, whether it's 46. I mean, I wouldn't ask you to draw 46 chromosomes, but understand that that's what's happening at the human level, because there's actually 46 chromosomes in the cell. Over here you have another example where a non-disjunction event occurred. You should be able to predict again how many gametes will be produced based on the error and what type of non-disjunction error it was. So what we'll do is we'll do this example together. You can try yourself first, pause the video, then come back and check your answer. So let's take a look over here. It looks like during metaphase 1, the chromosomes have the pairs have lined up, and then they're starting to separate. And ideally, all these would go to one side and all these would go to the other side. But that's not what's happening here. Here, we have an error. An extra chromosome is going to this side, and over here, we're missing a chromosome. So what we have here is a non-disjunction event in meiosis. There's only one S there. 1. And so if we have a non-disjunction event meiosis 1, that means we're going to have cells with N plus 1, N plus 1, N minus 1, N minus 1. And you can see that here. We have a total of 8 chromosomes. We should have 4 in each. 1, 2, 3, 4, 5, 6, 7, 8. Now, when I take a look at my cells that are splitting here, I have 1, 2, 3, 4, 5. I have 5 chromosomes. That means you're going to have 5 chromosomes in each cell at the end of this. So let me just draw my 5 chromosomes here. 5 chromosomes here. These are my N plus 1s. These are my N plus 1s. Whereas over here, I have 3 chromosomes, so I'm going to end up with 3 chromosomes in each once the sister chromatids separate. 1, 2, 3, 1, 2, 3. So these are my N minus 1s, my N minus 1s. Same idea over here. Pause the video, try it yourself, and then come back and take a look if you got the right answer. So it looks like metaphase 1 was good, anaphase 1 was fine. Equal number of chromosomes went to both sides. Now we have our two daughter cells here. This cell looks okay. So we would predict it would be, so there's 8 chromosomes, we would predict 4 by the end of this. And so we have 1, 2, 3, 4. The 4 are splitting equally, so that means we'll have 2 with 4 chromosomes each. So these ones are normal. These are our ends. But if you look over here, there's a mistake in meiosis 2. So there's a mistake in meiosis 2. An extra chromosome went to one side because the sister chromatid didn't separate here, and then there's one less on the other side. So you're going to have one that has an extra chromosome. So we'll have 1, 2, 3, 4, 5. 5 chromosomes in one. So that's our n plus 1. And then one that has a missing chromosome. 1, 2, 3. So instead of having 4, there's 3. And this is our n minus 1. I didn't draw the nucleus or anything around it. I just drew the chromosomes so you could see what the main point was. And again, if any one of these abnormal ones are used in meiosis, in the reproduction, then a zygote will be missing in this case, or having an extra chromosome in that case. So one of the thinking questions you could ask yourself is, which would you prefer? If a mistake were to happen in your meiosis, would you prefer an error in meiosis 1 or in meiosis 2? I want you to think about that carefully, discuss it, read up on it, and the hint I'll give you is to take a look at the gametes that are produced where the errors occur. So here we have our error in meiosis 1, here we have our error in meiosis 2, which is, let's just say, the lesser of two evils. So when you have an abnormal number of chromosomes, the term that we give to the organism is aneuploid or aneuploidy. It's not a multiple of n anymore, right? N is 23, a multiple of n would be 46. But when you have 47, you have 2n plus 1. It's no longer a multiple of 23 or a multiple of the n value. So there's different types of aneuploids, and the aneuploid is just the organism who has the abnormal number of chromosomes. There's trisomies, which have an extra chromosome, and there's monosomy, which are missing a copy of a chromosome. So usually when you have n plus 1, that results in a trisomy, which is 2n plus 1. When you have n minus 1, that results in a monosomy, which is 2n minus 1. So I'm going to teach you how to name conditions using the genetic nomenclature, and then you also have to be able to name the condition that it would be given for its more common name. So for example, for trisomy 21, its genetic name is trisomy. It's a trisomy. There's an extra chromosome, and then you have to indicate where that disorder is. So this would be called trisomy 21. Because it's on chromosome 21. And it's called Down syndrome. Now here, our 21 looks fine. Everything else looks good. I know there's a missing Y, but that's okay. That's because this individual is a female. She has 2 X chromosomes. But you'll notice that there's something missing here. There's a chromosome 14 missing. There's a missing copy. So this is a monosomy. And it's chromosome 14 that's affected. So we would call this monosomy 21. Monosomy 21. These are just some examples of genetic conditions that exist. So you can take a look at the table. You see that the chromosome number name is given to you. And then you have the condition name given to you, along with some traits and symptoms of the condition names. So you should be aware of these conditions, or any conditions that we see in our case study activity, and any conditions that are in your textbook. You should be able to spot a karyotype, be able to name the condition using the chromosome number and also using its more common name. I just want to point out that sometimes you might have trisomies or monosomies in the sex chromosomes. In that case, you would just list what sex chromosomes the individual has. So for example, this individual here has 3 sex chromosomes, an X, X, and then a Y. And so I want you to read up on those traits and symptoms and be able to describe these conditions and communicate these conditions on a test or an interview or a quiz. So you should be able to match these karyotypes here to the conditions on the previous page. And you should also think about whether these conditions were made by n+1 gametes, n gametes, or maybe some n-1 gametes. You should be able to characterize these as trisomies or monosomies as well. And again, tell me what their common name is, along with some symptoms and traits that you might see in the individuals. So here's an interesting graph that I want you to pay attention to. We have the age of the mother on the x-axis, and we have the infants with Down syndrome per 1,000 birth on the y-axis. And what I want you to notice is that something interesting here happens as the mother ages. As the mother ages, hopefully you can spot the pattern, the number of individuals born with Down syndrome increases. And it jumps up quite a bit. So as soon as you start going beyond the age of 35, 40 there's a bigger jump in that case. And it's most likely because there's higher chances of non-disjunction errors occurring. We're not 100% sure why. This is something that we're still researching, but there's more non-disjunction errors occurring in the mother as she ages. So at this point, when you're reproducing at an older age, there's higher chance of having infants with Down syndrome than if you were reproducing at a younger age, let's say around 30 or 25 or in your 20s. So here's some more practice for you to attempt. You should be able to figure out the genetic name and the common name. We'll do this example here together, and whenever you see a common name that's provided to you in the PowerPoint or in your readings, it's a good idea to read up on it and include it in your notes that you'll be showing us every once in a while. So here, let's take a look. I mean, chromosome 1 looks good, 2 looks good, 3 looks good, 4 looks good, 5 looks good. So does 6, 7, 8, 9, 10, 11, and 12. 13 looks good, 14 looks good. Here's our error. I mean, just do another scan again just to check to see if everything else is okay. Looks like we have a male here, good. But we have an extra chromosome at chromosome 15. So here we know that we have a trisomy, and it's at chromosome 15. So this is trisomy 15, and it's called Angelman syndrome. Here you can see a video of trisomy 15 and the traits and characteristics that the individuals with Angelman syndromes would have. It's a good watch and it's very informative, so I do encourage you to watch this. And whatever you learn from here, maybe add it to your notes in the abnormal meiosis section of your notes. So, like I said, sometimes you might have the normal chromosome number, but you might have an issue with the structure of the chromosome. So you might have 46 chromosomes, great, but sometimes because of an error that happens during crossover, the chromosomes might tear or break or add other pieces onto them, and then that results in chromosomal structure damage. So if you take a look here we have a chromosome pair, and if you look very closely, they should be the same length, but there's a big chunk of this chromosome here missing. So this can lead to some developmental disorders. And there's 4 main types of chromosomal structure damage. There's something called an inversion, a translocation, a deletion, and duplication. And I'll go over those main types with you. So I'm going to describe these types, and you can provide a definition as I describe them. You'll notice here we have a translocation that occurs, and that's when genes have swapped places. And this usually occurs when genes have swapped places between non-homologous chromosomes. Homologous chromosomes, that's normal, that's fine. But sometime a gene went from one chromosome to another, where it wasn't supposed to go, and that led to a translocation, which may lead to some developmental disorders. The gene's still there. It depends on what segment was removed and swapped. But the idea is that it's a swapping of genes between non-homologous chromosomes. The deletion is another possibility because of errors during crossing over. A segment of a chromosome may have been removed. So now you'll notice you have a shorter chromosome like we saw in that chromosome 6 on a previous slide. Sometimes a duplication can happen where a chromosome looks longer. So because of errors in crossing over and duplication, so on and so forth, now you have an extra copy of the gene. And having too much of a gene can lead to some developmental disorders as well, because the proteins that are coded by genes can have an effect on the organism. So duplication is essentially the opposite of deletion. Deletion, you lose the gene, so the chromosome becomes shorter. Duplication, you multiply, you add that gene, and the chromosome becomes longer. And sometimes you have what's called an inversion. And you can see that quite clearly here. We have our yellow gene or blue gene, and due to errors in the manipulation of the chromosomes during meiosis and crossing over, two genes or segments of chromosomes switch spots. Now that may lead to nothing, or it could lead to some damage as well, depending on where the switching of the spots happened. But you can see here that the genes have been inverted, they switch spots. And you can probably tell that some are easier to spot in karyotypes than others. So maybe it's easier to spot a deletion or a duplication than it would be to spot an inversion at a visual glance, at least. So on the following slides, I've included some examples of different disorders that relate to the different structural damages of chromosomes. So here we have our translocation, and I want you to read up on the different disorders that can result from translocation. So here we have an example of the XX male syndrome. There was a translocation that occurred where it shouldn't have. Like I said, usually the X and Y chromosome, they will pair up and they may exchange some genes. But one gene that they shouldn't exchange is the male gene over here on the Y chromosome called the sorry gene. But if that does happen, if they do exchange the sorry gene, it might go on the X chromosome. And what can result there is you might have a individual with XX that happens to have a sorry gene, sex-determining gene, which will trigger the development of some male characteristics. So when that happens, some things that you might see would be small testicles in the individual. Usually the individual is sterile, so can't reproduce. There might be poor facial hair growth. There might also be development of breasts on the male. Another example would be, well, if the sorry gene was lost on the Y chromosome, you might have a female that's XY, because during a translocation event, that sorry gene went to some other X chromosome in the other gamete, and this Y was passed on by the father, but there's no sorry gene, which triggers the male development. And that's all caused by a translocation. Sometimes you might have a duplication that happens. So again, here we can see the chromosome fragment has been repeated. And here we have a condition. I have some videos that you can watch in order to understand the condition in more detail. But we have Charcot-Marie Tooth disease type 1, also abbreviated as CMT. And that's all because of a duplication in chromosome 17. So usually the rule of thumb, but there's always exceptions, it depends on the gene, is that if you repeat a fragment 3 or more times, that's when you have more severe side effects or effects on the organism. Here we can see an inversion. Usually the effects are not as severe for an inversion because the genes are still there, just flipped around. But sometimes you can have some negative effects. Usually we've seen that there's a higher chance of cancer and miscarriages with these inversions. The most common inversion seems to be in chromosome 9. So just understand that inversions are not as severe because the genes are still there, just switched around on the chromosome. And then we have deletion. So a deletion again is where you have a fragment of a chromosome that's gone missing. You lost a fragment of the chromosome. And so you can usually see that because you have one of the chromosomes that's shorter. So if you have a deletion in chromosome 5, that leads to a condition called Cri-du-chat syndrome. And that just basically means cry of the cat. So if you look up Cri-du-chat syndrome and look up audio, you can hear what a baby with Cri-du-chat syndrome would sound like. There's a distinct sound that they make. So it's a very high-pitched cry. They usually have microcephaly, which means a small head. Cephaly essentially meaning your head. Micro meaning small, and they also have very low birth weight. So here I've included audio of Cri-du-chat. The link should still work. If it doesn't, you can just go on Google and research Cri-du-chat syndrome audio, and you can hear what someone with that sounds like. Here you can see chromosome 5, there was a deletion. And sadly, these individuals usually die in childhood. So now what we're going to do is we're going to talk about how to diagnose these non-disjunction errors that can happen. And usually what we do is we come up with a karyotype. So technicians can compare a karyotype by taking a sample of your white blood cells, not your red blood cells, and that's super important. It has to be your white blood cells. They'll usually add a few drops of a solution that will stop cell division in metaphase because that's when chromosomes are most condensed and therefore most visible. And then they'll take a photograph using their technology, and that spits out a karyotype over here. So you can see this happening. The sample of blood is being spinning around really, really fast so that you can get the white blood cells. You can then take those white blood cells, add your chemical to freeze the cell division in metaphase, and then get your picture of your chromosomes that are more condensed there. And then you can study that karyotype to see if there's any abnormalities with it. So if there's more than 46 chromosomes or less than 46 chromosomes or some kind of structural damage on the chromosome, you can spot that on the karyotype. I want you to think about why it is that you don't want to use red blood cells. Remember, the red blood cell is a specialized cell. You'll notice that it carries oxygen for you. When it undergoes specialization, it goes through this transformation where it ejects or spits out its nucleus so it can make more room to carry oxygen and have more hemoglobin to carry the oxygen. So hopefully in my little explanation there, you got a little hint as to why you don't want to use a red blood cell to make a karyotype and why you'd want to use the white blood cell instead. The white blood cell does go through a transformation, but it doesn't go through quite the same transformation that the red blood cell went through. One suggestion for women that are usually over 35, because of the increased chance of having babies with genetic disorders, is to do what's called prenatal testing, which is essentially where you test for genetic disorder prior to birth. And so usually there's a few different strategies you can use to do that. Sometimes the strategy might require cells from the embryo. So usually as early as 8 weeks, you can do what's called a chorionic villus sampling, a CVS, where you get cells from the outer membrane called a chorion that surrounds the embryo. So you can see that in this picture over here. You're trying to get cells from the chorion, which surrounds the embryo. And then what you can do is you can create perhaps a karyotype and see if there's any missing chromosomes, extra chromosomes, or any structural damage. Eventually, when the fetus is large enough, another test that can be done is amniocentesis, which is where you again, get cells from the embryo, but this time you get cells from the fluid-filled sac that surrounds the fetus, and that's called the amnion. So you can get cells from the amnion, and then again, the amniotic fluid rather, and then what you can do is analyze those cells, make karyotype, and again check to see if there's any missing extra or damaged chromosomes in order to judge if the child will have a developmental disorder. You'll notice that to do these tests, sometimes it can be, it looks quite scary. Yes, this is a very large needle that's used to go into the amniotic cavity to get the amniotic fluid in order to test these cells for some genetic disorders. I do want you to read up on these genetic testing techniques in a bit more detail in your textbook, and I do want you to add them to your notes. Typically when we ask you questions about comparing, contrasting, or making connections to the real world, these are great examples to speak about and to be able to describe, because it is something that is really out there, and it's a great application of biology. This slide is a comparison of amniocentesis and chorionic villus sampling. You can see there's a difference in time of when each can be done. Usually the CVS is done 8 to 12 weeks in, where you go and you get some cells from the chorion, and so you can see that happening over here. And then when the fetus is a little bit bigger, you can do another test called the amniocentesis, where you would go and extract some of the amniotic fluid using this big needle over here. Either way, you can do some biochemical tests with that, and you could also do some karyotyping. So biochemical tests might take a look at key signals, key molecules within the cells and fluid you extracted, and the karyotyping would allow you to see if there's any missing chromosomes, extra chromosomes, or damage to the chromosome that may have occurred, and that can give an indication if there's a developmental disorder that the child may be born with. Another example of a test that's done, a prenatal test, is the multiple marker screening. The advantage of this is that it doesn't really require cells directly from the embryo. You can usually do it between the 15th and 20th week of pregnancy. And what you do is you take blood from the mother and you just test for different hormones, and then the different hormones in the blood, the different levels, might tell you if there's a certain disorder that the developing embryo might have, for example, Down syndrome. Just another little point to mention is that usually prenatal testing is recommended for women that are over 35, heading into their 40s, because if you remember that graph we saw earlier, as you age, there's a higher chance of having children with Down syndrome, because there's an increased likelihood of making a non-disjunction disorder or error in your meiosis. So another very important concept to understand is that there are many couples that have an inability to conceive. They can't make a baby. So if you look in Ontario, there's about 10% of couples that experience problems with conceiving. They might just have reduced egg and sperm quality and count. The fallopian tubes that lead to where the fertilization needs to occur, they might be blocked. So there's many different reasons that couples can be having trouble to conceive, but there are some options out there that can assist them in conceiving, and those are called artificial reproductive technologies. We abbreviate that as ART. So I do want you to read up on those, especially in vitro fertilization and artificial insemination. You can see an example of in vitro fertilization over here, where we grab an egg from the mother, we grab some sperm from a sample that's provided, make the fertilization occur in a dish, and then afterwards, once that zygote has formed and developed a little bit, we can then implant it directly into the uterus. So that's just one example of an artificial reproductive technology. You can also see the example of artificial insemination, where we wash a sample of sperm and then inject the sperm directly into the uterus. So the main difference here is that we didn't create the zygote outside of the uterus. We instead injected the sperm in the uterus, where the zygote will hopefully be created once the sperm reaches the ovum. In the in vitro fertilization, vitro meaning glass, we created the child in a dish first and then implanted it into the mother that would hold it and allow it to develop in her, the surrogate mother. So here's our summary of what we've seen today in this presentation. We saw that there are errors that can occur during meiosis. Sometimes they can occur during meiosis 1, sometimes they can occur during meiosis 2. Those errors are called non-disjunction errors. If they occur during meiosis 1, your gametes are all abnormal. They are all either n plus 1 and n minus 1. But if the error occurs during meiosis 2, then you might have some chances of getting normal gametes, but you'll have half your gametes that are abnormal. One will be n plus 1, one will be n minus 1. And if you use these abnormal gametes, then that can result in abnormal number of chromosomes, which we referred to as aneuploidy, and the individual who has the abnormal number of chromosomes is called an aneuploid. And remember that we don't represent them by 2n, we represent them by 2n plus 1 or 2n minus 1. And again, the main point I need you to understand is that when your DNA is damaged or there's something missing, an extra chromosome or missing chromosome, you usually have some developmental disorders. So again, a trisomy is when you have an extra chromosome, monosomy when you're missing a chromosome. Over here you can see trisomy 21, Down syndrome, along with some symptoms. Over here you can see trisomy 13, which is Pateau syndrome, along with some of the symptoms and signs that the baby will have Pateau syndrome. Over here you can see monosomy X, which means there's a missing X. There's not even a Y here. There's no X. There's only one X, sorry. There's no other X. There's no other Y. There should be 2 sex chromosomes. And here we have monosomy X, and you can see some of the missing features, or the features of an individual with monosomy X. At this point, you should be able to do all the learning goals on the slide. And there's also textbook problems, which you'll see on the next slide. You should read the section, page 161 to 166. They pertain to all the concepts that we saw in this PowerPoint, and there's some good review questions that you should attempt. I do want you to read up on the artificial reproductive technologies in more detail, so that you should add these to your notes. So maybe make a table to compare the different types of reproductive technologies. And then at this point, you could even do your Chapter 4 review, Chapter 4 self-quiz, and we'll have a mini quiz on this PowerPoint in the next few days.